Hello everyone, so we're finally going to get ready to start talking about amino acids, something we've been working our way to talking about to better explain enzymes and proteins. So in the last video we talked about intermolecular forces, polarity, nonpolar molecules, things that we can use to build up to talking about the different amino acids and their R groups. And so our hope is to transition from amino acids to talking about protein structures to explain the active sites of enzymes, as we can see here with glyceraldehyde 3-phosphate dehydrogenase, and how important the amino acid cysteine is to its active site and reactions. Now, all amino acids differ in structure, size, and electric charge, but let's talk about some of the general ideas of amino acids. First of all, they have the same structural features. They all have a carboxyl group, an amino group, an R group, and a hydrogen bonded to the same carbon. The interesting thing about each of the amino acids we're going to study is that the R group can differ, giving the amino acid different properties. So we're going to start a little deep dive on the 20 most common amino acids, with first talking about the class of amino acids that are nonpolar and aliphatic. A quick review on the idea behind nonpolar molecules is that they have an even distribution with their electron density, as we can see here with nitrogen gas and the carbon chains I have as an example. So some of the R groups that we're going to see with these examples of amino acids are going to have an even distribution of their electron density at the R groups. There's a total of 7 amino acids in the common 20 that are nonpolar aliphatic, and these are the first 4. Now, one of the interesting things is that glycine is the only non-chiral amino acid, since it's bonded to 2 hydrogens, but it's the most simplest R group. As we can see with alanine, it's connected to a methyl group, and then it starts getting a little bit more complex with proline and valine. The last 3 amino acids that we have in this category are leucine, isoleucine, and methionine. The next class of amino acids we're going to talk about are the polar uncharged amino acids, meaning that these R groups have polar character, but they're not charged. Remember, the idea behind polarity is that one atom is pulling on the shared electrons so much that it forms a dipole, and one side of the molecule actually has a partial negative charge. First 3 examples of these amino acids is serine, cysteine, and threonine. I can't say the third one. Oh my goodness, okay. The last 2 being asparagine and glutamine. Now, the only difference between these 2 is the carbon length, so it can be easy to confuse the 2, so just try to pay attention. And for a little side note, it's going to be really important when we talk about disulfide bonds and cysteine later on in the next video when we talk about how these amino acids interact with one another. For the next class of amino acids we're going to talk about, these are negatively charged, so they actually have a full-on permanent charge. And there's 2 of them. We have glutamate and aspartate. Now, I know these look very similar to asparagine and glutamine, but we got to remember, with those, they have the amide type bonding. We don't have that with these 2. To couple with a negatively charged one, we have positively charged amino acids. So we have 3 of them, and here's the first 2. We have lysine, arginine, and lastly, we have histidine. Now, the interesting thing about histidine is that a lot of enzymes that we've looked at before for glycolysis and gluconeogenesis use histine residues in their active sites. For the last set of amino acids that we're going to talk about for the common 20, we have the aromatic amino acids, meaning that their R groups are aromatic. And we have 3. We have phenylalanine, always have trouble saying that one, tyrosine, and tryptophan. Now, the interesting thing is that tryptophan and tyrosine, and to some extent phenylalanine, absorb ultraviolet light. Now, I know I just listed all 20 of the most common amino acids and their properties, and that was kind of a lot, but let's talk about one more property of amino acids, the fact that they can act as both weak acids and bases. Remember, with the Bronsted-Lowry definition of acid and bases, a base is going to accept a proton and an acid is going to donate a proton. We can see here ammonia is accepting that proton and water is giving it up. You can actually see some of these properties of acids and bases in our amino acids because of the functional groups. In the carboxylic group, we can see that the oxygen can donate that proton and the amine group can accept that proton. So amino acids can act both as weak acids and bases, and because of this, depending on their local environment, is depending on whether or not they're charged. And this can be really important to certain enzymes. people refer to the isoelectric point, they're referring to the point, or the pH, in which the ion has a charge of 0. So I'm well aware that this video was really dense, since we just listed and talked about the 20 most common amino acids, but the point of this short and sweet video is that so we can build upon it in later videos when we talk about primary, secondary, and tertiary structures of proteins to help us talk about proteins, enzymes, and more. So I hope you guys have a great day.